Well, thank you, everybody. My name is Christian Boada. I am currently a postdoc here at Texas A&M College of Medicine. I work under Dr. Roderick Pettigrew in his lab. This is some of the work I've been doing for the last, I guess, 5 years. This was part of my graduate studies, and my focus is, or the focus of our lab is kind of biomimetic nanoparticles. And lately, what we've been doing is we've been developing nanoparticles that are lipid-based in their composition, but have surface functionalization from membrane proteins. So what we try to do is mimic as much as we can the lipid composition and the protein composition of different cell types to create nanoparticles that are still drug delivery agents and have, through these functionalizations, different kind of neat properties. And what I'll be presenting today is a specific apolipoprotein biomimetic nanoparticle, specifically for atherosclerotic plaque targeting. And I must mention as I begin that this work was really kind of helped by the nanosembler and is very much based on the nanosembler technology because it was kind of what enabled this process. As many of you know, the LDL is the natural kind of nanoparticle, and it's very interesting because what people normally know as bad cholesterol is really kind of this complex nanoparticle that kind of floats around in the body. You can see here a representation of this particle that's called LDL. And the LDL is kind of this macromolecule that has in here you can see the phospholipids on its surface, or you can see a protein component that kind of wraps around the whole nanoparticles, and then inside you have the cholesterol esters. This molecule measures around 25 nanometers and is the primary responsible for the accumulation of cholesterol within atherosclerotic plaques, and it drives the progression of what we normally know as a heart attack. And what is interesting about this is that, like I said, it's very much like a nanoparticle. It measures 25 nanometers across. It has very interesting kind of biological properties. It's able to talk to different receptors and develop, go throughout the body in very complex ways and target specifically. What happens is in a disease like atherosclerosis, you have kind of this overexpression of scavenger receptors. Now, it's kind of argued how it really begins. Is it first that you have a lot of LDL floating around? Does that drive the internalization? Does that create the inflammation that then drives the overexpression of receptors and then drives the accumulation? It's kind of debated, but the interesting thing is that they specifically focus on these areas where plaques develop, and these scavenger receptors are very different from normal receptors such as LDLR in that they're constantly being produced and they're constantly active, and that's why they're called scavenger receptors, because they don't really downregulate. Now, another thing that is interesting about the lipid nanoparticle, this LDL nanoparticle, is that it has only one protein. So from a standpoint, engineering standpoint, it's really kind of straight through. You have this nanoparticle that it's already available in the body. Why not just take the protein that kind of mediates the biologic interactions and kind of hijack it and use it as a drug delivery vehicle? And that was the reasoning behind what we did and the journey of creating this nanoparticle. And what we found out was that normally people really didn't like to do this nanoparticle or use this nanoparticle because it was very difficult to work with. To begin, the nanoparticle is pretty big. It's about 500 kilodaltons, has a very complex structure. It has many bonds, so it's not something that you can readily replicate in a recombinant system or express using bacteria. People have tried to express the different binding sites. However, it doesn't really replicate the whole complexity of this molecule. And we thought, let's try to use some of the methods that were developed in our laboratory and then go forward and try to functionalize this. And one of the key points in achieving what we wanted to do as far as functionalizing was really the synthesis procedure. Normally, what you would do is you use thin layer chromatography, which is this process of dissolving the lipids in chloroform, evaporating, then resuspending with water, and then extruding through a membrane to create a homogeneous kind of particle. Now that has the disadvantage of being something that, one, it's very labor intensive. Two, you actually have a lot of things. You have a lot of forces that are acting upon these nanoparticles. You have the extrusion force, which creates shear forces. You have heat, you have pressure. You have all these things that are really not conductive to creating or handling such a delicate nanoparticle. So this is where the nanoassembler came in. And the nanoassembler, I guess most of the people attending this would be familiar, is that you have these two inlets and they have the microfluidic chips, and through the mixing it comes together and it functionalizes or assembles the nanoparticle in kind of this quick step. Now that was kind of a blessing in disguise for us, because the micro mixing was actually gentle enough to to handle this protein, as I'm going to present later on. What we did is we took normal lipid formulations that you would try to make a nanoparticle, and then combine it with the protein, in this case, the LDL. What we did to procure the LDL, normally it's a process where you have ultracentrifugation over a period of, I think, a total of 24 hours. But we repurposed analytic technique of using fast liquid chromatography, using size exclusion to basically elude the nanoparticles. What we did is we took blood plasma, we ran it through this column, and then we would collect the different fractions as they come out. These fractions, much like you would hear, were selected on base of their absorbency. So we would take different peaks based on their protein or approximate protein concentration, I would say, based on this absorbance, and then collect every single fractions. To select the ones that we wanted, we first did a protein concentration to see where the protein was, and then our determinant, we measured the cholesterol concentration, and basically that would tell us where a majority of the LDL nanoparticles would be. And it turns out we collected these two fractions having the highest level of cholesterol, and then we use that to synthesize our nanoparticle. As we began to characterize this nanoparticle, you could see we compared two methods, the nanoassembler method and the TLE method. We took those two fractions that I mentioned before and went through the whole process of assembling it. And what we did is we measured the concentration of or the functionalization of the LDL protein, the B-100 on the surface using flow cytometry. What we did is we took samples of both of them and then incubated with an anti-ApoB-100 antibody with a chromatic fluorophore attached to it, and measure what the binding would be. You can see here, for example, the comparison between these two. The liposome groups as a control. You can have the nanoassembler APO13, which means that this was made with the APO13 fraction. The functionalization is 71%. And then the other fractions that we chose, which is the fraction 12, made with the nanoassembler, was close to 41%. In contrast, when we did the same thing with the TLE, you can see here that it's less than 20%. It looks like there's a lot of aggregation. It wasn't really conductive. And when we did the same thing with the 12, there was even less, so 10%. As a control, and not shown here, we did also pure LDL, and the pure LDL was close to 80% of the total population of events that would be positive. So you can see here, using the APO, the fraction 13, we were able to get a lot of surface functionalization. This is, you can see here a little bit, is the ultrastructure of our nanoparticle. We compared here, we have just your normal liposomes, and then you have your LDL particles, and then you have the best fraction that was selected from the flow cytometry analysis, which was APO13, and they seem to have closer resemblance to these liposome formulation. Our physical characterization analysis, we saw that the size was around 91 nanometers, and the surface charge was very close to neutral, while still getting a dispersity index that was very, very favorable. When we did other protein analysis on the surface of this nanoparticle, including STS, you can see here that we have the band characteristic at, again, this is a very large protein. So we have the band that is almost 500 above the limit, which is 250 here, but we have here the mark of the pure LDL. We have other proteins, however, we weren't able to do the full proteomics on this, but we also need to mention that, especially for the plasma fraction 13 and the aposome, we have protease mix. So it might have been some of those things that are showing up here. You can see also the comparison when we have the whole blood plasma just flowing through compared to the pure LDL. And the nanoparticles, you can see that the liposome has absolutely no proteins on the surface, while the aposome has a signature that is very close to the fraction 13, meaning that there's very good functionalization on the surface. When we took these samples and did a Western blot, we can see that the aposome was positive for that ApoB-100, again, corroborating that we're able to actually have this at least on the surface. On the APO, we wanted to test the efficacy of kind of our synthesis procedure. So we took a common contaminant, which was ApoB A1, which is one of the HDL proteins, if not the only HDL protein, if I remember correctly. And you can see here that there's very little signature other than the whole blood plasma, meaning that our synthesis procedure was able to kind of remove most of the proteins that were in the blood plasma, effectively purifying the LDL. Now, this is also a drug delivery vehicle, so we also tested the encapsulation. For this particular study, we chose rapamycin just because this is something that we use for testing currently in their lab for its application towards atherosclerosis. You can see here that when compared to the aposome, the encapsulation efficiency was really not that different than a normal liposome. And when we did our release curve, comparing to rapamycin, we can see that it's a prolonged release curve reaching about 80% at 6 hours. When you compare it to the liposome, which I apologize for not labeling, but it's the circle. The liposome are these circle plots on the graph, and the aposome are these square, sometimes triangle plots on the graph. You can see that the release is slightly higher at each time point, meaning that the aposome does release a little bit quicker than it does for the liposome. And we hypothesize at least that this is due to the fact that the rapamycin is actually loaded on the lipid bilayer. So if you have such a large protein like you have for the LDL, they're going to be sterically competing for those for that space on the lipid bilayer. You can see that the difference is significant. Regardless, you're able to see that it's still a prolonged release curve. And moreover, that the aposome does retain this drug loading capability for rapamycin. In the future, we'd like to test more compounds, but it was, at least from that perspective, it was encouraging that we were able to load rapamycin at a similar rate to liposome. We took this nanoparticle and we injected it in vivo, and we did this because the biodistribution of the nanoparticles is actually very important to us, and then also it's hard to replicate in vitro systems. So beyond safety, we really kind of went straight in to the animal model. And for this animal model, we had ApoE mice, which developed plaques after 12 weeks of a high-fat diet. And we injected them with rhodonamine labeled liposomes and aposomes. And then we had an in-phase preparation, which is the dissection of the aorta. And then we looked at these plaques under the confocal microscope. You can see here the bright field images, the white is the actual plaque, how it looks when you're looking at it. And then the fluorescence, you can see here that the blue is the collagen around the plaque. And then the black spaces are usually where the collagen has broken down or the plaque is developing. When we see the liposome, you can see that there's very little red signal corresponding to the rhodamine liposomes. Meanwhile, when we see the aposome, we see this incredibly bright red spots all over the plaque, meaning that there's accumulation within the plaque. What was remarkable was that we saw that it was accumulating not only at the edges, but we were able to see also in the middle, which is a hard area to penetrate when you're talking about atherosclerosis, just because the fibrotic core is often is not often targeted enough in atherosclerosis. We would something interesting happened when we were looking at this, when we were looking at these animals. We wanted to know a little bit more about where the co-localization was for the nanoparticles. So we injected a marker for CD68. It was a CD68 marker labeled, I think, with another fluorophore. I can't recall at this time. But as soon as we injected that, when we were looking at the rhodamine channel, it completely killed the aposome accumulation on the nanoparticles. And you can see the contrast is actually very, very notable. And as you can see here, when we actually look at the numbers, you can see that the liposome was very low. The aposome had very high cases, and then also due to the scale, some very low cases, but overall still higher. And you can see that the average increase in accumulation was 20-fold. And then you can see when we inject the CD68, then that goes back down to almost liposome levels. So from this, it was interesting to know that the accumulation was very much dependent on a CD68 pathway. And to be perfectly honest, we don't really know what this is. CD68 is not really a marker that you're normally associating with LDL internalization, although it belongs to that family of receptors. Of course, we would like to see investigate this further in the future, but it was very interesting to see how this mechanism was responsible for the internalization of these nanoparticles. And moreover, and what would like to be the takeaway of this is that we increased accumulation of these nanoparticles over the plaque by 20-fold when compared to liposomes. This, for us, at least for the future, we would like to think that a targeted approach towards atherosclerosis would be very much beneficial in increasing the efficacy of available drugs such as statins, such as rapamycin, which is often limited by target effects. So we're thinking that if you're able to deliver specifically to these plaque areas, you're not only going to be able to increase the efficacy, but also reduce the toxicity. So for the future, we would like to try some compounds where we go and we test, are the effects increased by the incorporation into a specialized and targeted drug delivery system, such as the aposome. Another factor that we tested that was very important to us in determining kind of the application of these nanoparticles was actually the compatibility. There are some nanoparticles that are known to have immunogenicity upon repeated exposure. And for us, especially dealing with proteins and proteins derived from patients, is it going to matter if you inject to another patient? Is it going to matter? Is it going to develop antibodies against itself after repeated injections? So what we did is we took mice, we injected them once a week with a dose that once a week with a dose for 4 weeks. The total dose of these 4 injection was 100 milligrams per kilogram. Then we sacrificed the mice, spun down the blood, and collected the plasma to collect the antibodies. And then these plasma was put in together with nanoparticles to see if they would bind. We tested IgM and IgG just because these two are responsible for the acute and chronic response for antibodies. So if there are antibodies that would develop after these repeated injections, we would be able to see by the binding on the nanoparticles. When we had the comparison between liposomes and aposomes, you see that neither of them, the IgM or IgG, had a significant increase in antibody binding on the nanoparticles, meaning that the nanoparticles didn't have very high immunogenicity. Moreover, the nanoparticles were made with human ApoB protein, and we were injecting in mice. So if we were able to, if we had anything, we would have been able to see it. This attests to the aposome's capability and to our whole biomimetic approach. The biomimetic approach in itself has the advantage of normally we're able to see very high, very favorable toxicity profiles, and we don't have to worry about is it going to kill the cell, is it going to kill the animal. So that really helps out, and this is very much proof of that. Uh, what I would like to kind of for the big picture process here is the comparison of the synthesis procedures. Now, me as I was developing this as a graduate student, frankly, I was hoping that the microfluidic method would be the one that I would eventually end up using just because it was so much easier. And you can see here just the comparison of scales is crazy. The purification of whole dose plasma would take for us at least 15 minutes. If we were doing TLE method, the ultracentrifugation would maybe take another 26 hours just to get the whole plasma. Instead, with the FPLC, we were able to narrow it down to 30 minutes. And then where we were doing thin layer evaporation, the process was very involved, very time consuming, very user dependent also on how skilled someone was at handling the machine. It would take 6 hours. And like I said, it's very labor intensive. You're subjecting high heat, high pressure to the proteins all the time, and you're able to see that through the poor ApoB enrichment. While the microfluidic synthesis, you got this in under 30 minutes, and you were able to get 72% enrichment on the nanoparticle. So it was really something that for people that we were collaborating with at the time, it was something hard to grasp because they were so much they were used to this TLE method and how it usually made. But I have to say that the microfluidics really was remarkable and is pretty much the only synthesis procedure that would accommodate this type of synthesis and these type of functionalizations of such a large protein. With that, I would like to end and thank everybody for listening to this. And if anybody has any questions, I'll be glad to. Thank you.